In this topic, we're going to discuss transpiration and the factors affecting the rate of transpiration. So we're going to look at the water movement through a plant. This is just recapping as we've covered this in a previous lesson. Why plants wilt, how you measure the rate of transpiration, the different factors that affect transpiration. So you've got temperature, humidity, air or wind movement, and water supply. So just to recap, water movement through a plant begins with evaporation from the leaf surface. Remember that 98% of water taken up by a plant is lost to the atmosphere by transpiration. So the stomata allow for the diffusion of carbon dioxide into the leaf for photosynthesis. So when the stomata are open, water can move out of the leaf. So water evaporates from the spongy mesophyll cells and then it diffuses out of the stomata down the water potential gradient. Remember that transpiration is the movement of water vapor out of a leaf through the stomata. Now, transpiration is useful in plants because it helps to keep water moving up the xylem, and evaporation helps to cool the leaves. The next step is that water moves up the xylem to enter the tissues, so it moves down its water potential gradient. And water moves up the stem in the xylem due to the tension caused by the water lost from the leaves. So we discussed that in the previous topic. It's called the transpiration pull. And water is taken into the root hair cells by osmosis. So let's have a look at wilting. Why do you think more transpiration takes place during the day compared to at night? Well, this is because the stomata are open during the day and they're closed at night. So the stomata are open during the day so that carbon dioxide can diffuse into the leaf. Remember that carbon dioxide is the raw material for photosynthesis, and it diffuses through the stomata into the chloroplasts inside the mesophyll cells. The stomata close at night to reduce the volume of water lost by transpiration. The stomata may also close in hot, dry conditions during the day so that water lost by transpiration is reduced. So what is wilting? Well, if the plant doesn't get enough water, it's going to start to wilt. This is because its cells have lost so much water that they're no longer turgid or full of water. So turgid cells are firm and they give the plant support. If the cells become flaccid, then the plant becomes soft and the stem no longer stands upright. And the leaves start to droop. Now, wilting's not a bad thing. The leaves move downwards so that they're out of the direct rays of the sun and so they don't get as hot. So when the temperature decreases and the plant's able to absorb more water than it is losing water by transpiration, the leaves will recover. Okay, let's have a look at how to measure the rate of transpiration. So the rate of water uptake can be measured using something called a potometer. So you need to label this picture on page 6. Now, we need to know how quickly water is lost by transpiration from a plant and what environmental factors make this process go faster. It's not easy to measure the rate of transpiration, but you can use a potometer to measure the rate of water uptake. Now, something to consider, the volume of water lost by a plant is slightly less than the volume of water taken in by the roots. This is because some of the water is going to be used up in photosynthesis and in keeping the cells turgid. However, in this case of the potometer, we do make the assumption that water uptake is equal to water lost. So labeling this potometer, you've got the leafy chute that's placed in the apparatus, and it's placed in the apparatus underwater so that there are no air spaces. There's an air bubble that you look at. Okay, so the rate at which the bubble moves can be recorded by measuring the distance on the graduated capillary tube, and because it's rate, you also measure the time, so you measure the time using a stop clock. You've got water sitting in a reservoir. This is used to set the apparatus back to the beginning. So when you open the tap, water flows into the apparatus and the bubble is pushed back to the starting position. Okay, here's a potometer that's a little bit more fancy, but the principle's the same. So you've got the graduations on the left, and these are used to measure the water uptake. The syringe on the right is used to reset the apparatus between the readings. And here you have a very simple potometer. So we use a similar apparatus in class. You can colour the water so that it makes it easy to see it moving up the graduated capillary tube. The tube can be refilled by doing another reading. The tube can be refilled to do another reading by squeezing the rubber tubing, and this sucks up more water from the beaker. Okay, moving on to factors that affect transpiration. Can you think of a few factors that are going to affect transpiration? Well, the rate of transpiration is going to change as environmental factors that affect the rate of evaporation change. So these can be investigated using a potometer, and you keep all other factors constant except the one that you're changing. So for example, factors that will affect transpiration include temperature, humidity, wind or air movement, light intensity, and water supply. Okay, let's have a look at temperature first. An increase in temperature is going to increase the kinetic energy of the water molecules, so this increases the rate of evaporation of water from the leaf. So how would you vary this in an experiment? Well, you can use an electric heater or an environmentally controlled room. So fill this in your notes. If you were to draw the graph of the results, you'd have temperature on the x-axis and rate of transpiration on the y-axis. Notice how, as the temperature increases, the rate of transpiration increases until a certain point where it plateaus. This is because as you're increasing the temperature, there's an increase in the kinetic energy of the molecules so that water evaporates faster. Then the curve starts to plateau. This is because another factor is becoming limiting. Now, there might be a point where the rate may even decrease because the plant has stopped photosynthesizing and the stomata closed to conserve the water. Okay, the next factor is humidity. If you increase the atmospheric humidity, what do you think is going to happen to the rate of transpiration? Well, this is going to decrease the diffusion gradient between the leaf and the atmosphere. So, it's going to decrease the rate of transpiration. So here you can see two leaves exposed to different environmental conditions. On the left, you've got dry condition. On the right, you've got humid condition. Notice how there's a steeper diffusion gradient on the left when the environment is dry compared to on the right when the environment is humid. So when you've got a steep diffusion gradient, the rate of transpiration will be faster. So how can you vary the humidity in an experiment? Well, you'd place a plastic bag over a leafy shoot that's attached to a potometer. Then you need to take a number of readings as the humidity in the bag increases. The results should show that the rate of transpiration slows down as the humidity increases. The next factor is wind or air movement. If you increase the wind, this is going to blow away any moisture around the leaf so that there will be an increase in the diffusion gradient of water between the leaf air spaces in the atmosphere, so that the rate of diffusion of water vapor will increase and the rate of transpiration will increase. How do you vary this in an experiment? Well, you would use a desk fan at different speeds blowing on a plant that's in a potometer. Now, water vapor moves out of stomata and it creates what we call diffusion shells around each stoma. This creates a gentle diffusion gradient. Now, air movement is going to blow away these diffusion shells that form a gradient of humidity around the stomata. This makes the diffusion gradient between the air chamber and the atmosphere steep. And it increases the rate of transpiration because water vapor diffuses out quickly. Now, some plants have got small hairs on the lower epidermis, and these stop the diffusion shells being blown away so that they conserve water. If you were to draw the graph of rate of transpiration against the air movement, remember that air movement will go on the x-axis. So as air movement increases, the rate of transpiration will increase until it reaches a maximum. At this point, so where the graph starts to plateau, all the stomata are losing water as fast as possible. Okay, light intensity. Plant stomata open in the light so that they can take in carbon dioxide for photosynthesis. So the rate of transpiration is going to increase as the stomata open. So an increase in light intensity will cause an increase in the rate of transpiration. So how do you vary this in an experiment? Well, you would set up a light source about a meter away. Wait 5 minutes for the plant to adjust to the new conditions and reach the maximum transpiration rate. Now this is quite important. You do need that time for the plant to adjust. Then you take 3 readings, record, and you find an average. You move the lamp closer and you do the experiment again, and then you continue to move the lamp closer. So this increases the light intensity. So if you were to draw a graph of light intensity against rate of transpiration, what do you think it's going to look like? Well, the light intensity is going to go on the x-axis, the rate of transpiration on the y-axis. So as the light intensity increases, the rate of transpiration increases. This is because more stomata are open to bring in carbon dioxide for photosynthesis. Then it's going to start to plateau. So after a certain point, the light intensity is not going to affect the rate of transpiration any further. This is because all the stomata are fully opened. Okay, lastly, water supply. If a plant is short of water, the stomata are going to close. So the rate of transpiration will decrease and the plant will not be able to photosynthesize when the stomata close. In summary, we looked at the movement of water through a plant, why plants wilt, how you measure the rate of transpiration, so remember how to set up a potometer, and then we discussed the different factors that affect transpiration. So we looked at temperature, humidity, air or wind movement, and water supply. And that concludes our lesson. The end.